Good morning, ladies and gentlemen, for those of you who are present here in South Africa, and good whatever time of day it is for those of you online. Thank you so much, all of you, for coming. This is another very exciting meeting of the mRNA Technology Transfer Program. Really a sort of progress report to see where we are. And where we are is, as you'll soon find out, really amazing. I think what's been done in such an incredibly short time is truly wonderful. So my name's Charles Gore. I'm the Executive Director of the Medicines Patent Pool. And we are, along with the World Health Organization, supporting this program and are very honored to be doing so. And first of all, I'd like to invite Anban Pillay from the Department of Health in South Africa to give some opening remarks. Anban, the floor is yours.
Good morning, everyone. And good, as you say, morning, yeah, midday or evening. I'm not sure where people are. But it's great to be here. And welcome to South Africa and Cape Town. I hope the weather improves. Cape Town weather can be great during summer. It's really a great pleasure to be talking to you at a meeting on tech transfer. Very often when we're talking about tech transfer, we're often debating whether we're going to agree on tech transfer. And here we are, not just talking about agreeing on tech transfer, but actually doing it. And here we're having a progress meeting on that. I think it's important to appreciate how we got here, because if we forget how we got here, we may often lose our way as we move forward. We had huge challenges in terms of our COVID-19 pandemic, not just South Africa, the continent as a whole, in terms of production, access to vaccines, medicines, PPE, etc. And I think the world has realized that it's very important that developing countries have capacity to be able to produce, and technology transfer in terms of vaccine production, and I'm hoping that that extends to other areas of medicine production as well over time, allow many developing countries to become self-sufficient, not just for the pandemic, but I think for other diseases that affect us as well. So we're very excited that there's good progress in terms of this area of work, and I'm glad to see so many participants from across the globe that are here today, as well as online, and I'm looking forward to hearing as well from them on the progress made. So from the Department of Health side, welcome, and we hope that this is going to be a fruitful meeting for everyone, and we're very excited. Thank you.
Thanks so much, Anban. And now I'd like to invite Sophie Bell, who's the Consul General of France, to Cape Town. And France has been very instrumental in this entire program right from the beginning. And in fact, it came out of something that France said during COVID about the need to increase manufacturing in low and middle income countries. And a vaccine task force was set up under COVAX. And the program is one of the things that came out of that. So,
thank you, Mr. Gore, for your words. Good morning, everybody. It's a pleasure and an honor to be with you. I also want to greet the members who are joining us online. It's very important that you are all here and represented. It is really my pleasure today to introduce this three-day meeting jointly with our South African partners. As you know, and as you have mentioned, France and what we call Team Europe has been strong supporters of the mRNA Technology Transfer Program since its inception in 2021. And France, right at the beginning, took a very strong political stance, saying, you know, we need to share this technology with developing countries. They need to benefit from it, and hence, let's develop production capacity in Africa and other countries. So France has dedicated 70 million euros to this program, implemented by the World Health Organization and the Medicines Patent Pool, and is very proud to be the first donor of this program. I am also delighted that this meeting is taking place in Cape Town, since we have the technology transfer hub here in Cape Town, and I want to greet the South African consortium members, and especially Biovac within the room. Looking back at where we started 3 years ago, in the midst of the COVID-19 crisis, I think we can collectively be proud of what has been achieved. As this event aims to take stock of the program and its progress and look at how we can further leverage our win to accelerate its impact, let me share with you a couple of key learnings from these past years. First of all, the mRNA technology transfer program is evolving in a crowded global health landscape with many competing political priorities. There are more than 30 regional production initiatives under development on the African continent alone. So the first lesson is that we need to coordinate and better connect all these initiatives to avoid competition and rather find complementarities between them. This is why European Union partners have come together under the flagship Team Europe initiatives on manufacturing and access to vaccines, medicines, and health technologies in Africa. This is a program called MAV+. I'm sure you've heard of it. We hope that the mRNA program will strengthen the capacities of industrial partners in low and middle-income countries to develop production chains which will benefit from the financial incentives offered by the Accelerator for Vaccine Manufacturing in Africa that was launched in Paris last June in partnership with the African Union. This is what we mean by connecting existing initiatives together. The other strength of the Team Europe approach is a close cooperation with the African Union and its health agencies under a joint EU-AU health agenda. This dialogue offers a platform to obtain the political buy-in from all partners so that emerging countries can support the new production chains by choosing to buy health products manufactured at the regional level. And I really want to stress that point. I think it's another lesson that we have learned over the past 3 years. Without the commitment of governments who are benefiting from these projects, the production capacity won't grow, and ultimately it will lead to failures. So we will really need the full commitment of the different governments to really bring this new industry to a stage where it can, thrive and grow. This is very important, and I think we all have to be very careful about this. The second lesson is that the mRNA technology transfer program is evolving in a fast-changing public health landscape. The program was able to demonstrate a high level of agility to adapt to partners' needs in terms of R&D pipeline. This was made possible thanks to flexible funding envelopes. This gives us all a lesson, if still needed, on how a program which was initially tailored to respond to the COVID-19 crisis in 2021 was able to pivot to become relevant to a broader set of diseases beyond COVID-19. Thanks to this approach, the capacities which are built through the program are leveraged to address other health challenges that are more relevant to the target populations. The third and maybe last lesson for now is that education and training is a central piece of the technology transfer puzzle. It is critical to see technology transfer not only as an investment in technology and equipment, but also as an investment in the people. The adequate training of human resources is essential for the full appropriation of the mRNA technology platforms by the program partners. Going forward, we will need to further develop South-South cooperations to accelerate the exchange of learning between partners in low and middle-income countries. And we believe that the MPP has a key role to play in promoting this approach. The WHO Academy, which will be launched in France in Lyon in December on the 17th, will provide all partners with a platform to connect and share training materials, including on technology transfers. To conclude, let us keep in mind that while the mRNA technology transfer program supports emerging and developing countries with the development of regional manufacturing capacity, we should not lose sight of the end goal, which is equitable access to health products. And this end goal should never be undermined. I wish you insightful discussions along these 3 days. Let us keep this critical agenda moving to the benefit of local and often underprivileged populations. I thank you.
Thank you very much indeed, Sophie. And now I'd like to ask our panel to come up. And to start with, Martin Frieder is going to give you an overview of the program's achievements. So Karen and Imelda, if you'd like to come up. So Martin is the lead at WHO on this program.
Thank you, Charles. So it's great being here, but I'm going to take you through a bit of a walk of history. So we were here in September 2021, standing on the lawn of the French Embassy in Pretoria, and we announced that we were going to start the mRNA program. This was a result of the COVID pandemic and the fact that the low and middle income countries were not getting access to vaccines, and especially not getting access to the most advanced, the mRNA vaccines. And we'd been asked by the member states to do something about this. And we looked at the technologies. We said, well, what do we use? Do we do the inactivated viruses? Well, we could do that, but if you've built the technology, what else can you do with it? Nothing. We could do the viral vectors. Well, that's nice technology. We've got the chimp adeno. But if you do that, what else can you do with the technology? Nothing. And we'd had the previous experience of building pandemic influenza vaccine technology around the world. And we'd realized that if your technology cannot be used for other things, then it quite rapidly becomes obsolete and your facilities go bankrupt. So the choice was mRNA, because mRNA, in theory, can be used for many, many disease targets, infectious and non-infectious diseases, and should therefore be sustainable. So we set this up, and I want to remind you what our objectives were. It was to provide to the existing manufacturers in the low and middle income countries, provide them with mRNA know-how, the ability to make mRNA vaccines. But also, just as importantly, was to build new technology where there was new vaccine production capacity where there was none. So let's get in and look, what have we actually achieved? Can I have the first slide, please? Who's doing slides? Oh, okay. So, okay, there's a button here. Okay, technology evades me, you know. Right. So, this is the overhead, and the rest of the conference, the rest of this meeting will be looking really at this data. So just to see what have we done and where are we going. So in 2020, there was no mRNA know-how in any of the LMICs at all. We've got a total of 15 partners, plus Afrigen, that makes 16. And so in 2020, there was no RNA capacity at all. Now, at the end of this year, 2 of them will be able to have mRNA vaccines being produced at GMP level. That means going into the clinic, going into manufacturing. And by somewhere between now and 2030, there will be another 9 added to that. So this is progress that is moving forward. It would have been nice if we were going faster, but these things take time. And so you can see going from 0 to mRNA vaccines research and development. We will have most of the partners doing R&D by next year. and then more than half of them being GMP ready by 2030. Now let's go on to the next slide. So let's break this down in more detail. So here you can see this by our various WHO geographic regions: Africa, Eastern Mediterranean region, Southeast Asian region, Western Pacific region, European region, and the Americas region. And you can see here now in gray, it was those that began with no vaccine capacity at all. And we can see that we've got one of them going from no capacity at all to being able to produce mRNA at research, going on by 2025 into GMP manufacturing, one in Africa. Then we've got the same in the Americas, one that already had some vaccine production that will be doing GMP manufacturing next year. But the main message here is that by 2030, most of the partners will be producing GMP, and all partners will have moved forwards, even if they're not able to get to GMP. This is an important message. So this program has already made significant progress, and in the next year or two, we'll be making even more significant progress. So the reason that we set this up was, first of all, pandemic preparedness, that all of these countries need to be able to respond to a future pandemic. So what is going to happen if the next pandemic will take place? We just don't know when. So if the pandemic was to happen in 2029, what are the various scenarios? Well, the one scenario is that actually all of our partners basically are only producing very, very small quantities. Now, there's a reality in life. There is no such thing as a free lunch. Well, there might be today, but otherwise there is no free lunch. And so the real challenge is going to be that if governments are not going to be procuring anything at all, what we're thinking is that actually these manufacturers will have really small manufacturing facilities, really small capacity, and if nobody's buying, they will be able to produce together about 1.1 million doses. And this is on a model where they just produce 1 or 2 batches per year that they provide. Now, we've seen this being done in the US, where the US government procures pandemic influenza vaccine every year, and they then pour it down the sink. So that's that model: buy and pour it down the sink. Now, the next model is actually there is a pandemic, and the government tells the manufacturers, please provide us with pandemic vaccine, and we will buy it from you. Now, if they've still just got these small small instruments that we're currently recommending them to buy because the cost of maintaining them is low, but you can still get GMP, they'd be able to provide about 60 million doses. But with this know-how, they can quickly, in a matter of months, scale up to be able to provide about 2 billion doses per year of pandemic response vaccine. That'll be, so let's cross your fingers, no pandemics between now and 2029, please. Whatever you can do to avoid that. Right, so now let's look at what this means in terms of response time. Your pandemic starts on January the 1st. As you all know, that's when pandemics start. We're just recovering from the night before, and your boss calls and says, Martin, get your butt back to the office, there's a pandemic. So what is going to happen from our manufacturers in the 12 months following this pandemic? And here we can see the curves, which are looking at the different kinds of scenarios: a low case scenario, mid case, high case scenario. This is a function of either how many micrograms of RNA we require in the facility, or whether we've got manufacturers that have got small, medium, or bigger case investments that have been done. And we can see that in the 3 scenarios, can we go back? Oh, that way. Here, from 2027 to 2029 to 2034. And at 2034, this is when we start seeing really potential things at 2 different scenarios: 100 microgram scenario, which is what we're currently working, 30 micrograms and 10 micrograms. And that we could be getting up to 600 to 200 million doses within this kind of production scale, where we've got 12 months over here. So these countries will now be able to respond to a pandemic with the mRNA capacity. But the caveat to all of this, and this is what much of the rest of the discussion is going to be, is they were built on the premise of a COVID vaccine production. Today, there is no market for COVID vaccines. So for all of these facilities, what are they going to do to stay alive? And much of the rest of today and tomorrow is going to be around discussing that. So since many of you here are funders, are donors, I thought I would just quickly look at what this is doing. Now, let's go back to the past, the pandemic influenza. We saw that for every dollar that was given to WHO. The WHO invested in pandemic capacity in the countries, and we had a total of 13 countries at the time. The national governments and the regional development banks, etc., invested a total of $17. So for every dollar that came from the WHO facility, $17 came then from national facilities. So this shows what catalytic investment can do, that WHO with MPP, when we say this is a good project and we are putting money on it, the governments come behind and say, well, if they're doing it, we will follow suit. And so here we can see in Afro, this is for every dollar that's been put in by the mRNA tech transfer program, this is what other sources have put in. You can see the catalytic effect here. In the Latin American regions, this is primarily the national government putting this in. Southeast Asian regions, that's the national governments putting this in. Now we've got some that are still waiting. And what we're hearing is the waiting is saying, yes, but what else are you going to produce? And that's why we're here. Now, since you're donors as well, what about investment gaps? So mRNA, it takes money. So it's quite easy to put a small laboratory research facility that costs you, it's a couple of $100,000. But it is building this ability to transition from a non-GMP laboratory research to a GMP, small-scale GMP. That's what we're talking about here. That's about 12 million, I think they've got that, 13 million USD per partner, is what it costs to build something which is a small GMP facility. And this translational, going to translate from the university bench to a manufacturing capacity, this goes through this step of having a pilot facility. And this pilot facility, this is what is missing in most of the low and middle income countries. They've got manufacturing, that's great. They've got research, that's great. But they do not have the capacity to translate from research to manufacturing. There is this translational gap in the middle. And so this is the kind of investment that we're looking at to fill this gap. And that is it from me. I think this, in a nutshell, I hope, captures what we will be talking about over the next 2 days. So if you understood that, there's coffee outside. Charles, back to you.
Thank you very much indeed, Martin. And next, I'd like to invite Ike James, who's the head of the MPP's technology transfer team, to give an overarching technical report. Ike.
Thanks, Charles. Thanks, Martin. Good morning, everyone. Great to be in Cape Town. The next few days, we'll really look forward to hearing from our progress, from our partners in terms of their progress, their challenges and developments. That's where the real meat lies. I will give a brief overview of where we are, complementing on Martin's message, where I'll provide a technical overview of the achievements to date. So just to complement on Martin's feedback, if we rewind back to 2021, amidst the COVID-19 pandemic, the mRNA technology transfer was established to address equitable access in terms of vaccines, technology, and know-how. As Martin said, the mRNA technology was selected above other technologies due to several advantages in responding to outbreaks and pandemics. And the South African consortium was formed with the concept of receiving a technology transfer of an already established platform. This did not realize, and so the consortium needed to embark on its own journey to develop mRNA technology that was for the program. This mRNA technology would be made available to the 15 manufacturing partners through multilateral technology transfer. Sorry, just going to the next slide. Through multilateral technology transfer with the aim of strengthening local production and future pandemic preparedness. The program partners would then have the opportunity to further develop the technology, select disease targets relevant for their region, and build on a product pipeline through knowledge sharing and collaborative development, and therefore supporting sustainability during interpandemic periods. The multilateral technology transfer approach has really worked well in this instance, considering 15 manufacturing partners, all unique in their own way, all requiring different levels of information at different times. And so it was really important to maintain engagement, and the key here was to understand the gaps and needs for each of these partners, and allow them early access to information so that they could build their laboratory capabilities, allow them to familiarize themselves with the technology before receiving full tech transfer. So in terms of the achievements to date, if we look back at when the program was established in June 2021, within 6 months, an mRNA technology suitable for discovery was put in place. In March 2022, just 3 months later, Afrigen had put together an introductory training package that allowed the manufacturing partners to travel to Afrigen and receive training on site. In December 2022, an mRNA vaccine manufacturing process was established, representative of the process that we have today. And in May 2023, the Biomanufacturing Training Hub in Korea was put in place to support the training of staff in the program. And to date, we have a couple of hundred staff that have actually received training from IVI in Korea. In June 2023 was actually a pivotal moment where we had immunogenicity and efficacy data supported by our preclinical studies. In December 2023, the BioVac facility was fully modified to allow for a multi-platform technology, also a pivotal moment, and we'll hear from BioVac a little bit later today on their efforts in that. In January 2024, the mRNA process at Afrigen was further scaled up to a 1 liter IVT process, and we'll hear from Karen. Similarly, in Jan 2024 as well, the regulatory capacitation and strengthening project was initiated with the South African National Regulatory Authority, SAHPRA. In August 2024, the mRNA platform was ready for transfer to all of our partners, and 13 on-site gaps and needs assessments of our partners was completed. In September 2024, so 2 months ago, the mRNA platform at Afrigen was ready to transfer to all of the partners besides BioVac. And October 2024, we received toxicology data showcasing the vaccine safety. And now we are at initiating the technology transfer to all of the other partners on besides BioVac. We have completed 3 already. Currently, Synergium from Argentina are currently on site at Afrigen receiving the technology transfer training. So we'll hear more about them later in the program. So in terms of technology transfer to the manufacturing partners, this is just a snapshot of where we are from comparing December 2023 to December 2026, where we plan to have completed the technology transfer demonstration and technology transfer received by our manufacturing partners. So to date, December 2024, we would have completed 93% of all on-site gaps and needs assessment. This will be completed next year. The technology transfer readiness plan to date stands at 50% complete. So this is basically each of the manufacturing partners identifying what they need to do, what they need to have in place to be able to absorb the mRNA technology. This will be completed next year, this time next year. The technology is then to be transferred from Afrigen. To date, we're at 29%. Next year we would have completed the remaining 10 manufacturing partners. And then in terms of technology demonstration and transfer completion, we have at 7%, with this being completed at BioVac. Next year, December 2025, it'll be at 50%, and in December 2026, it'll be at 100%. This is building on what Martin was saying earlier, is what do these facilities do once they're established? And last year at the WHO Cape Town meeting in April, the manufacturing partners were asked, so what diseases are relevant for your region? And this is the profile that came up. And so it was then observed that it's really important that product pipeline becomes a core focus of this program, being able to build this. And so when we look at what we've done, last year we held a meeting in Southeast Asia where we brought together governments, academic institutes, development organizations, and manufacturing partners in Southeast Asia. And we really had a brainstorming session to identify and select disease areas relevant for development. And there 4 R&D consortia were established. One for dengue, one for Plasmodium vivax, malaria, therapeutic HPV, as well as hand, foot, and mouth disease. This was really successful, and so it was used as a blueprint to identify disease targets relevant in other areas. And so earlier in October, we had a meeting in Brazil where we identified 2 new R&D consortia, one being influenza based on seasonal and pandemic response, and the other being leishmaniasis. We have in plan to do this in the East Mediterranean region in Q1 2025, and an Afro region plan for Q4 2025, with the understanding that building this pipeline for the region is of critical importance going forward. Also worth mentioning, it's not only disease targets that is important to focus on, but we also understand that mRNA technology is dynamic and keeps on evolving. So understanding new technologies that would further reduce the cost of goods, thermostability, freedom to operate, etc., are all key components in staying relevant in this field. So this brings me to my last 2 slides in terms of the overview. So in terms of key achievements to date, if there is a message that we can highlight, we can say that the program, an mRNA Technology Development and Transfer Center has been established in South Africa. An mRNA technology platform has been established and is ready for technology transfer to all of our manufacturing partners. We have a network of manufacturing partners globally, 2 in LATAM, 6 in Africa, 2 in Europe, and 5 in Southeast Asia. The technology transfer of this established platform has been initiated. A network of R&D consortia has been established, and we will continue to grow these consortia. We have a global training hub for biomanufacturing, as we understand that human resource skills development is a key component to this. And regulatory strengthening project has been initiated because this is a key component in establishing an end-to-end vaccine manufacturing ecosystem. So these are the achievements to date. And in saying that, we've had the wonderful support of our donors. We have mobilized $123 million of funds as of November 2024, with funding both to the South African consortium and catalytic funding to our program partners. Without your support, none of this would have been achievable. We aren't done yet. There's still some way to go, but the future is looking very positive. Thank you.
Thank you very much, Ike, and it gives me great pleasure to introduce Karen Fenner and Imelda Jordan, who are from Afrigen, who are going to talk about various things, including the sort of pivot from COVID to a more sustainable approach. So, are you doing a double act, or are you doing one after another?
It's a double act.
Oh, good. Even better.
So, thank you, Charles, Martin, and MPP and WHO for giving us this opportunity to present the progress. As you can see, this is a joint presentation Imelda is the head of quality and regulatory, and I lead the tech transfer, development tech transfer streams in Afrigen for mRNA. Okay. So these are the topics that we are going to cover today. But I'd like to take you on a journey in this development from inception to where we are today. You've seen this slide many, many times, but it's really significant for us. Short people. It's really significant for us because it shows how the WHO, with the MPP and all the partners, were able to mobilize a network for future sustainable pandemic preparedness and vaccine equity in just 12 months. So Ike already showed you a roadmap, but since the hub was announced, we've had about 3 years, and in the time that it took, about 6 months to get contracts in place, budgets approved, and so forth, within 6 months, we were able to show the first lab-scale mRNA vaccine. And this has really been wonderful in the collaboration with Professor Patrick Arbuthnot and Chrissy Bloom and Abdullah Eli at Wits University, who helped to fast-track the learning. And so we had a lot of pressure to package our learning and transfer this to the partners as development was occurring. The first partner training, as Ike said, started in March of 2022. I can't see the slides over there, so please forgive me for turning. And that was with Synergium and Biomanginos. And at the same time, roughly, Afrigen entered into a research collaboration agreement with the NIH, and the Vaccine Research Center. And this was ready to fast-track pDNA development, which is the starting material for mRNA, and also GMP manufacturing, as well as working together on different priority disease targets. We had the first mice immunogenicity study completed by June of 2022. So this was the first evidence, early evidence of AfriVac 2121. And we continued with scale-up work, and the first 100 ml batch was completed in December of 2022. So the material from this process was used for the first hamster challenge study. Ike has already mentioned it, in May 2022. And this was done in collaboration with the University of Marseille, with Antoine Nougharet and his team. Our team was very privileged to be able to go to the VRC when the GMP batch was being manufactured for plasmid DNA. This was part of the tech transfer and training, and we're very grateful for the US government support. So by November of 2023, we've had 14 of the 15 partners come to Afrigen for hands-on training. The start of year 2 was marked by technology transfer demonstration batches at the 1 liter scale with BioVac. And at the same time, we were also able to lock the DS process at the 1 liter scale, which is the final scale. A significant moment for Afrigen was when we received the filling line, which took about 13 months to design, manufacture, qualify, and then come to Afrigen. And then, as Ike mentioned, a huge milestone for us was when, through all the different scale-ups, we were able to take that material and put it back into mice studies at the larger scale, confirm protective efficacy in hamster studies again, and also to show that the vaccine is in fact safe. We also had our first publication recently, now in September, and the team has managed to do phenomenal work in developing 11 out of the 14 assays, which have been transferred, aren't being transferred to the partners. We are currently doing manufacturing technical batch 4 with the view to lock the DP process. And then by the end of the year, we will confirm the stability of AfriVac 2121, not only in physicochemical readouts or in vitro readouts, but also back in murine immunogenicity studies. And we will do additional technology transfer demonstration batches and hands-on training with 3 other partners, totaling 4 for this year. Okay, so at the heart of this program is building capacity. And if you look at this value chain, which is the South African vaccine development value chain, from discovery all the way to early development, you can see all the key players that are listed down here. And it really includes from genomic surveillance all the way through to the clinical platform development. Ike also mentioned that we are working on other technologies. So in terms of the formulation platform, we also have discovery of lipids with Charles de Quinning and Robin Clintworth. We have enzymes that are key reagents in the IVT that are being developed by Professor Heinrich Fulschkenk and his team, and obviously further bioprocess development, which is happening at SIBA at UCT. So working very closely on the ground, on the development, we were able to identify areas that needed strengthening. And I'd just like to highlight 2 of these today. So in the space of preclinical screening with the challenge studies, through our collaboration with the University of Marseille, able to transfer protocols and know-how to the University of Cape Town with Dr. Roz Chapman, who is now conducting hamster challenge studies. And then in terms of GLP toxicology, so Afrigen contracted Agile Life Sciences in India, and through that partnership are able to further support a local GLP toxicology hub, which is being led by Rose Haishi and her team, and they are doing a pilot study at the moment. And so we'd like to also thank the SAMRC for making this possible with funds. Okay, so this is how we organized the work packages according to this breakdown structure. And ultimately, validation of the platform would be through success in the clinic. So looking at what are the key dependencies for that is a clinical trial approval. And so we organized our work packages as you can see here. So preclinical studies with stability, all the development reports for drug substance and drug product, these form work packages together with everything that is needed for a GMP certified facility to make clinical trial material. All of that forms part of an internal technology transfer package, which is the same as the transfer package that goes to our partners. And so now I'm going to hand over to Mulder, who's going to elaborate on the GMP readiness and manufacturing.
Good morning, everybody. Can I just say, behind this road to success, there's a very hard-working team that you will find in Afrigen and supported by the rest. But let's just give you a big view of where we come from and where we are now. We will be ready with our mRNA manufacturing facility, the GMP certification next year, and we are currently also very busy with our pDNA development adjuvant and the automated system that will be the next rollout of our certification. But just a quick look in where we were in 2021 with the mRNA manufacturing facility and where we are now in current status. And this is the Unit 6, which is the mRNA manufacturing unit, if I can introduce it to you like that. When we started in 2021, you'll see at the side, in 2022, we requested SAPRA to do a facility readiness assessment, of which they've identified that our quality control laboratories will need major adjustments, and we had to move out our research and development team out of one particular unit, move them into Unit 9 and Unit 10, which you will see tomorrow. And we had to redo quite a lot of our units, which was 5 and 7. So the real handover to the R&D team was towards the middle of 2023. They moved to the new unit, and we could then use that particular unit and for all our laboratories. So the final handover of the facility from the engineering team was early in 2024. And therefore, we are moving as fast as possible to get it up to at the GMP certification level. So whilst we were talking about units, I can just, let me see if I can. There. This is the Unit 5, which you will find the full finish right at the back. That is the Grade B area with the visual inspection areas, packaging here, and the adjuvants already planned into. This is the Unit 5, as I stipulated. We did find out that during last year we could not find a contract packer that can help us with our drug product bulk to be packed. And for that reason, at that stage, we were GMP, and for that reason, we moved to a full end-to-end facility from PDNA right to a full final drug product. This is the Unit 6. This is where the PDNA manufacturing unit, the mRNA formulation into a final drug product, with the micro-laboratory, analytical laboratories, our micro-laboratories, stability room, cryogenic facility, and with their PDNA design unit right there at the bottom. This whole area is approximately 200 square meter end-to-end facility and cater for all aspects of a fully end-to-end facility. Can I just give you a little bit, and you will see this unit tomorrow, just to give you a little bit of insight. If you look at the HVAC system, due to all the changes, you will find that the HVAC unit, that is for fresh air and heat and controlling the moisture, the ducting alone is 220 square meter that we had to fit into the above the manufacturing unit. And that can give you an indication of what a spider web of activities on the roofing. That does not even talk about the water for injection, which is built on a skid of only 15 square meter, and it to produce 200 liters per hour to facilitate the plant, giving it to 19 points and runs more than 400 meters on the roof. So just indication the complexity that was that unit, to have such a small unit and yet all the services of a big area. Right, then, everything that we are busy doing at the end-to-end development and the manufacturing is actually solidly built on our quality management system, which means that all the document management, change controls are managed, deviations are managed, CAPAs, the corrective action and preventative actions are well controlled within the quality management system. And that's account for all areas, from the plasmid design into the master cell banks, plasmid manufacturing, our analyticals for drug substance and the drug product, as well as the full finish. And I think something that's very unique about Afrigen is the fact that we do provide our further detailed more analysis. I want to give you just a very peep that you will see on some of the video material at Afrigen of the state-of-the-art equipment in the drug substance and the drug product. Now, due to space, I think there are a lot of the some specific equipment, which was more pilot scale, had to be almost re-engineered to make it more GMP approved. And there I must say that our engineering team worked very closely with some of the manufacturers to do the necessary adjustments to get it to a GMP level. Then on the final product, you'll see our full finish equipment that we waited for quite a while to get there, but and it is the final stages of performance qualification at the moment. And you'll see that, and also the visual inspection areas are there. And let's look at the capacity of our facility. The mRNA drug substance current batch is 1 liter. Bulk drug product that the team are working on is between 3 and 6 liters. And then if you look at our full finish machine, the volume that can be filled, the filled volume can vary from 0.2 mils up to 100 milliliter, with the accuracy up to 0.5%. We can deliver, the full finish unit can deliver 1,500 units in an hour. And that, if you extend that over a period, it can manage almost 50 million doses in a year. Also very importantly for us then is our regulatory strategy, and that we need to ensure that we are in line with where our regulatory, South African regulatory team are working towards. And for that reason, we ensure that we engage with SAPRA in an early stage of product development to ensure that our processes are clarified, all the requirements, the material that will be produced, and ensuring that we address potential issues and are in line with their expectations. And we foster transparent relationship with SAPRA, and we do collaborate with our fellow industries with regards to regulatory requirements and needs. We do partake in a lot of the regulatory workshops to ensure that we are well informed, and we do also in line with a lot of our other groups, and also focus on continuous development and the training of our staff with related to regulatory requirements. That's all from my side. Thank you, Karen.
Now for the fun part, the data. Okay, so our strategy for validating the mRNA platform was to perform preclinical comparability studies of Afrivac 2121 and compare it to approved commercial COVID-19 vaccine, and to complete the manufacturing and the analytics data packages to conclude the tech transfer to the partners. The drug substance process development work began at the tiny 200 microliter scale, and this is a schematic that shows you all the scale-up work that has been done to the final scale now at the 1 liter in vitro reaction. Very nice to see that we've been able to maintain similar yields as we have scaled up the process. Similarly, for the drug product process development, starting at just 1.5 milliliters, as Imelda said, we have gone to the 6 liter. We have pushed the Knawa nanoscaler benchtop machine to its maximum, I think. But I've worked closely with Knawa and have done quite a lot of process development, and we will lock the process in at 3.6 liters. But we do have the ability to further scale that if we need to with some further time. And so this are the key steps in the manufacturing process with the different unit operations. And from one batch, we can produce about 24,000 doses. This will be at a 50 microgram dose. In front of you, you see the drug substance and the drug product analytical profile. The team have developed de novo, we all know that word, from the very beginning. These that are highlighted in orange, so a significant amount of work. And what that means is that there is deeply embedded know-how and expertise that is now being transferred to the partners. When we change out to new variants and disease targets where there's a different gene of interest, it is just these that are highlighted in orange that changes, but the rest all remain unchanged. This is a summary of the preclinical comparability data. So we have completed all of these and indeed have confirmed that AfriVac 2121 is safe, it is efficacious, it is stable, and we've been able to validate that through the preclinical work and therefore the mRNA platform. A summary of all the preclinical studies that have been done. What remains outstanding is to confirm immunogenicity after the engineering batch. Currently, we also have a non-human primate characterization study that is on the go. It will end at the end of this year. And the data from that study will be used to further support the negation of the need for NHPs. As it relates to the mRNA platform. As you all know, we will not take AfriVac 2121 to the clinic due to the changing COVID-19 epidemiological landscape and the lack of commercial interest. However, we need to continue building the capacity, so the team continues to build a mock CTA and continue our learnings through the process, which will also be very important that we are ready for the next disease targets. So in terms of some of the data, I'm going to skim through this because of time, but I feel really important to say that all of this is built on a foundation of rigorous scientific rationale and data, which supports the validation of the platform. So what you see in front of you is the dose-dependent induction of binding antibodies, where we show comparable activities between our vaccine as well as compared to the commercial vaccine. And this work, I'd like to acknowledge Professor Penny Moore and her team who have worked alongside us in this. We were also able to show comparative neutralization activities when compared with Pfizer and also with Moderna, as well as dose-dependent T-cell responses, so showing robust spike-specific CD4 and CD8 responses and no real difference between our vaccine and that of the commercial vaccine. And here I would really like to acknowledge Professor Wendy Berger and Roanne Keaton for the work that they've done. And so I mentioned that we had our first publication, so we've done 2 hamster challenge studies, both at the 100 ml scale and at the 1 liter scale, which has really shown that our 100 ml process is in fact representative, and the data is fantastic. So I encourage you to seek out the publication online, but we've been able to show that AfriVac is in fact protective and comparable with the commercial vaccine. The GLP toxicology study has shown us that our vaccine is indeed safe. There have been no adverse events, and we are very grateful to Agile Life Sciences and the contribution of PATH, who have been supporting us in this work. And so how transferable is this mRNA platform to variants and other disease targets? So here I can show you that we have been able to show comparable yields and integrity or purity between XBB 1.5, which is a variant, and the index strain, which is Wuhan. We've also been able to show that the drug product is able to meet all the draft acceptance criteria, and it is not different to the Wuhan strain. So in keeping with the South African tradition, the big 5 in Afrigen, as we call it. These are our priority disease targets that we are working on with our partners. I would like to encourage you Yes, they're all still in early development, but come and visit us on tomorrow. We'll be able to meet some of the scientists working on this, and then listen out for the talk by Dr. Amin Khan, our Chief Scientific Advisor, who will give us a talk on innovation and sustainability for mRNA, and then Dr. Pia Stegler, who will give us some insights into the route to scale up and the clinic for the TB vaccine. And while I'm on this, I want to just give a shout out to all the Afrigen team who's listening online. Okay, so Ike has spoken extensively about the tech transfer program, but how is Afrigen contributing to this to ensure pandemic and interpandemic sustainability? This is a summary of all the work that we have been doing towards the tech transfer. So as I mentioned, we have developed the process at the 100 ml scale as well as the 1 liter. And there will be 14 methods or analytical methods that are transferred to the partners. And I think very important to mention is that the level of detail and the quality of the document packages are the same, whether it's at the 100 ml scale or the 1 liter scale, which is important, and also the level of detail in the hands-on training also remains the same. So 2 of the partners will receive the 1 liter and then the rest at the 100 ml scale. And so yes, we have started the technology transfer demonstration batches at Afrigen. So the first partner back into our facility has been the Torlak Institute from Serbia. And how lovely, you can see them holding the drug product that was manufactured while they were on site. We also currently have the Argentinian team in Afrigen, as Ike mentioned. Synergium, you will hear a little bit more from them later on today. And there they are. You can't see, but they're holding their drug product, which is really significant for us. And then what is upcoming, we look forward to welcoming the next partners, which is India, Biological E, in early December, and then Indonesia, Biopharma, in February. And so Afrigen remains absolutely committed and dedicated to ensuring that all the knowledge transfer and expertise gets passed on to our partners, and we would not be able to do this without the diverse, unique partnerships that we have and all the funders. So I'd like to acknowledge all of you today. I won't name you, but we are indeed grateful. Last but not least, I must single out the Afrigen team for their dedication and their commitment, their can-do attitude. It's really unparalleled. Thanks.
Thank you very much indeed, Karen and Imelda, both of you. Sorry, can you get in? We've actually got a coffee break now, but I'll take sort of 5 minutes. If anyone's got any burning questions, you can obviously talk to any of the panel during the next couple of days. But if anyone's got any questions now, I don't know whether we have a roving mic, but if not, if you just speak loudly and I'll... Oh, you have? Okay, perfect. I will ask you a difficult question, Karen. You talked about 2 things. One, the amount of stuff that you've done de novo. And secondly, the can-do attitude of Afrigen, which has been amazing. Have you ever got to a point where you've gone, I'm not sure how we're going to do this? Have you ever felt stuck? Or have you always thought, no, so that's what I meant by a difficult question.
Is it a trick question?
No.
I think that, yes, we have felt stuck at times, but we have had, as I mentioned in the last slide, amazing support from so many around us. And I think that for the team, in those moments of feeling stuck and just maybe a little bit despondent, because that does happen, as Pietro always says, that I always say, eyes on the prize to the team, and I think that they have felt such a dedication to the big picture and knowing that they are contributing to something that is going to have a global impact. And so that has kept us going and motivated. And let me say one extra thing, which I think sometimes we forget to mention, and that's because MPP technical team tech transfer team has been with us in the trenches. So I just really want to say thank you to Ike and the rest of the team who have provided us with technical support in this process.
Thank you. I mean, that's in some respects great to hear, because this is not easy, and what you've done is really amazing in a very short period of time. And I would have been very surprised if you'd gone, no, we never had moments like that. So, that's really good. And to some extent, there may be moments like this for all of the partners when you're developing things. But it's just about, exactly, eyes on the prize, just push forward, and there is an answer there.